Get started. So Ben, go ahead.
Yeah. For those of you that don't know me, I'm Ben, or the other Ben. I work as Ben's assistant director for Ask. And just to give a brief introduction for Dr. Göpfrich, in her undergrad, she attended the University of Nuremberg, went on to complete her PhD at the University of Cambridge, and is currently working on her postdoc at the Max Planck Institute for Medical Research. So her work is consisting of combining DNA, origami, microfluidics, and 3D printing to assemble synthetic cells. From here, I suppose I'll let Dr. Göpfrich continue to explain more about her research and everything she does in her lab.
Cool. Yeah, thanks. Thanks so much for the introduction, Ben. Let me share my screen. Is that okay? Can you see the presentation? Perfect. Yeah, actually, I moved on. I moved on since I started. A good year ago, I started my own group in Heidelberg. After I'd been to Stuttgart, and it's just now been over a year, so it's an exciting transition. And yeah, so all of a sudden, instead of just having my two hands to build a synthetic cell, I have many, and that's obviously super cool to work together with lots of talented and young and motivated students. Yeah, I thought I structured my presentation like this. I'll tell you a little bit about what we do, what all of this is about, but then I also want you really to give you the time to discuss the topics you would be interested in discussing. So I made a slide overview with a few topics that we could tackle. I want to leave it a bit up to you what you want to discuss about, but first of all, I'll give you a brief, say 10 to 15 minute introduction to our research, and then we can talk about other things as well. Yeah, I think in general, science is about solving puzzles, right? And there's so many exciting questions. And for me, one of the most exciting ones is whether it will be possible to build a cell from scratch. So to assemble a synthetic cell from its molecular components, really to ask the question whether it may be possible one day to achieve a transition from matter to life in the laboratory. So if you think about it, every cell that we encounter on Earth today is emerging from a pre-existing cell, right? There has to be some point in the history of our planet where actually life emerged from non-living matter. So doing this in the laboratory, like actually building a cell from its molecular components, something that can reproduce itself and evolve and show all the characteristics that we know from a living cell, that would be my dream, let's say. And it's not just my dream. There is an entire field behind this question. It's synthetic biology. And if you hear about synthetic biology, maybe some of you will think about Craig Venter, who was tinkering with a bacterial cell and its genome, stripping down a number of genes to make it simpler. So actually, this is one way of doing it, but this is not quite what I mean, because it's not really synthetic, right? If you take a pre-existing organism and you tinker with it, you alter it quite significantly. It's really impressive work. But what I really mean is a transition from so basically a bottom-up assembly of a synthetic cell, where you isolate biomolecules like proteins from a cell and you recombine them really with the vision to achieve a transition from matter to life. And you can imagine that putting these pieces of the puzzle back together is an extremely challenging task. And many people are working on this, trying to isolate molecules, trying to recombine molecules inside compartments and so on. It's super hard, and actually nobody knows if it's possible. And I think because it is so hard, we have the freedom to be creative. So in my group, instead of using biomolecules from cells, we actually make our own synthetic building blocks and program their assembly to really make new cells, synthetic cells, with new ways of information storage and replication. So they may look a little bit different from life as we know it, but the idea is that they have the same basic functionality. Why would you do this? It may sound even more complicated than this approach. Well, I think in bottom-up synthetic biology, it's a bit like you're working a bit like an archaeologist, right? You're trying to piece the pieces of the puzzle back together, and this can be super hard. And actually, the best you can do is as good as the original. So our approach, I think of it like this. You use new tools, you use new materials to achieve something that has the same functionality, but actually getting there may be much more straightforward, and you can incorporate maybe superior elements or other elements at least. So if you do this, the question is, of course, what tools and what materials could you use not to make a vase, but to make a synthetic cell, right? And I think, first of all, it's super important to say that there's no reason whatsoever to be dogmatic about it. So we are currently screening the horizon for new and upcoming things, for new tools, new materials, new technologies that we can actually use and incorporate in synthetic cell assembly. The latest addition to our toolbox was actually the 3D printer. So right now we're using direct laser writing to really arrange components inside synthetic cells, to use a laser to basically print structures inside the compartments. But for now, I want to introduce one tool and one material that we are already quite familiar with. First, the tool that we've seen is very, very handy for synthetic cell assembly is microfluidics, and the material that I want to introduce to you is DNA nanotechnology. So let's start with and then I want to show you how we implemented one very important function, namely the division and the growth of synthetic cells, probably one of the most fundamental aspects if you think about living cells, that they have to be able to replicate, right? So let's look at microfluidics. So microfluidics has actually developed lots and lots of on-chip functions independently of synthetic biology, but we can directly take them and use them for our purpose. So first of all, there are modules to make cell-sized compartments. So simply by intercepting a water flow coming from the left with an oil flow, and this video is slowed down 1,000 times, so this is super high throughput. Once we have a compartment, we can then inject components into these compartments, biomolecules or synthetic components, and also grow them. You see, after injection, they become larger. Then there are other modules which we can directly use to divide the compartments on chip, and we can then sort them according to specific properties as a kind of an artificial selection mechanism, and then finally release them in a native environment or also 3D print them in a specific pattern. So these modules are really useful, and when you see them playing together here on this slide, these are all things that or on-chip modules, microfluidic modules that we built in the lab, and I think you can really envision an automated assembly line for synthetic cells, and this is exactly what we want. So it's a really useful tool, but of course a compartment and its manipulation is by far not everything. So of course we need content, we need materials inside, and here I think that DNA nanotechnology is super useful for now, because actually folding proteins and predicting protein structure from the sequence is A, more expensive, and B, it's by far not as easy to predict protein folding compared to DNA folding. So we really use DNA nanotechnology to make shapes. So DNA nanotechnology in general is about repurposing DNA as a construction material. So it has nothing to do with genetics. It's purely about architecture. Architecture based on the complementary base pairing of DNA, the fact that A binds T, C binds G. And using this, you can imagine that you can build things. The simplest thing that you can build from a piece of DNA is simply a linker. So imagine you want to link component A with component B. Then usually a very, very straightforward way of doing this, especially for us physicists, is actually to use a DNA strand, because you can link almost anything because of the broad toolbox for chemical functionalization for DNA is already out there and it's commercial. So you can link anything from small molecules to proteins to nanoparticles, cells. You can also tune the linker length from angstrom to micrometer. You can tune the stiffness of the link. The linkage is programmable, and it's also reversible upon basically any stimulus that you like. So like this, you can already build a connector. And if you think about it, like nature is not always using the most straightforward way to link two components, so this is already stripping down complexity. But in the end, to build really functional parts, linkage is often not enough. And because of this, we use a technique called DNA origami, where you take a long single strand of DNA, you know its sequence, So you can design short pieces of DNA which bind in specific positions on the long strand and pull it together. And if you have many of these short strands, like 200 or so, then you can really fold desired shapes like this crane or this crocodile. And what the transition showed you is that actually the clue that we need for synthetic biology is, of course, dynamics. And this is exactly what you can get by using a different set of short DNA strands. You can reconfigure these shapes. And this is how you can really build dynamic and functional architectures for synthetic cells. So how do you visualize them? Well, actually, these DNA-based architectures are mimicking, or we use them to mimic proteins. So they are just as small as a protein would be, meaning they are on a nanoscale. So actually, we use an atomic force microscope, as you can see it over here. It's basically an arm with a fingertip. It scans across the surface, and once it touches an object, it gets deflected, and this deflection can be measured, and that you can see in a height profile. So for instance, here you see the iconic smiley face that was done by Paul Rothman in Caltech in 2006. So yeah, this is an architecture built completely out of DNA using this DNA origami technique. And today, actually, the whole process has become very much streamlined, which is why it is so convenient. So you simply download a software which is called CAD Nano. It's basically, some of you may know AutoCAD from 3D design. So this is just a nano version of it. You literally just draw the shape that you want, and the program can calculate the sequences that you need in order to make that shape. So, and then you simply get an Excel sheet with a list of the sequences, and you can send it off to a DNA synthesis company, and then all that you need to do is literally to mix the different sequences and heat them up, cool them down slowly, and then you have billions of crocodiles or smiley faces or whatever you designed in just a drop of water. And of course, these are somehow proof of principle shapes, right? They are not functional, but we have in the past used them to really make functional shapes. So on the left, you see simple DNA linkers that we used, for instance, to build a contractile cytoskeleton. So we used actin, and we simply, actin, so the natural cytoskeletal protein, and then we simply linked it with DNA. And then upon illumination, and we release ATP, so the chemical energy, and then we can really contract, and the DNA linkers really can guide the contraction towards the place where we want it. So that would be a prerequisite for motion, for instance. You can build reversible structures that attach and detach upon pH. And then you can use the DNA origami technique to really build functional structures from DNA. So for instance, here, these plates that you see here, they are quite a boring shape, just a square, but if you absorb them on the membrane of a compartment, so basically on our synthetic cell compartment, then you can really see that you can stiffen the compartment, that you can form kind of a cytoskeleton made from DNA. I've also made lots of DNA nanopores. So if you think about it, like ion channels, of course, sit in the membrane of each and every single one of our living cells, right? In the cell nucleus, we have the nuclear pores, which transport RNA out of the nucleus. In the cell membranes, we have other ion channels and transporters that allow for communication between cells, for instance. Yeah, here we build artificial ion channels, because actually ion channels are one of the components that are super hard to isolate from living cells. And there we have great potential if we can actually make them from scratch. Plus 50% of the drugs that we're currently using are targeting ion channels. So if you can make your own ones to replace non-functional ion channels, this also has an application, I would say, in the context of biology. Yeah, for me, these were always components of synthetic cells, of course. Yeah, so you see, we've been able to build like compartments. We've been able to build some functional structures for synthetic cells. The question is, of course, what is missing? And very obviously, if you want to build a synthetic cell, it has to be able to grow, and it has to be able to divide. So we put this very high up on our agenda to implement synthetic cell division. And actually, if People in synthetic biology have tried to reconstitute proteins now for decades to achieve synthetic cell division with the proteins that divide living cells. But this has never been successful so far. And this, I think, again, gives us the freedom or the right to be creative and to seek other solutions. So we were thinking, can we divide synthetic cells actually relying on simple physical principles? Well, the first thing we came up with is what I showed you in the beginning. It's kind of a splitting system, a microfluidic system where you make 2 compartments out of 1 and 4 and so on. But this is not really autonomous. So this is not really what we want. So we thought, okay, let's go back to the drawing board. What do you actually need for division? So first of all, you need to somehow define the plane of division, right? You need to tell the synthetic cell where it should divide. Actually, we thought, this we already have, right? Lipids can phase separate into a liquid ordered and a liquid disordered phase. So we made these vesicles, which are composed of 2 hemispheres. And then we thought, okay, how can we now achieve division? Well, what else do we need? Well, somehow we need to change the surface to volume ratio because 2 small compartments have a larger surface compared to their volume to compare it to 1 big one. So how do we change the surface to volume ratio of our synthetic cell compartments? Well, by osmosis. So these membranes are permeable to water, just like cell membranes are. And then by evaporating some of the water in the background, you can increase the osmolarity, and then these cells will divide and diffuse apart. So this works really well. And because it's based on a simple geometrical mechanism, we can build a geometrical model, a parameter-free, completely analytical model where we describe the synthetic cells as 2 hemispheres and basically put in that the volume change equilibrates the osmotic pressure and that the deformation is minimizing the contact area between the phases because the phases simply want to demix. So it's energetically not favorable for them to be in contact. So and what this model gives us is predictions that we can test. So it tells us that, for instance, the osmolarity ratio that we need to require to divide synthetic cells is square root of 2. So we can test this. And then it also tells us that any process, prediction 4 here, any process that increases the osmolarity should lead to division. So we'll just look at prediction 1 and 4 now. We'll skip the other ones. So osmolarity ratio required for division is square root of 2. So we prepare buffer solutions with different osmolarities, different numbers of particles in solution, and we see that at 1.41 square root of 2, these things really divide. Then we say, okay, prediction 4, any process that induces a sufficient change in the number of particles in solution should be suitable to trigger division. So we thought, okay, can we actually use an enzyme, a metabolic reaction? Can we feed these synthetic cells with a sugar and then add the enzyme invertase, which is eating up the sugar and thereby like doubling the number of particles in solution in each reaction step? Can we achieve division with that? And actually in this video, you see that exactly is happening. So bit by bit, the constriction forms and 1 cell becomes 2. We can also gain complete control over the process by using photocleavable components, which you can split by illuminating them with a laser, 405 laser, for instance, 4 or 5 nanometer laser or UV light. And then you are also changing the number of particles in solution. And this now gives you even the opportunity to select one lipid vesicle for division and the other ones remain unaffected. So you can really change this locally even. So then after division, of course, you need growth. And of course, we went back to our favorite tool, DNA nanotechnology, to build some linkers which can fuse. So we feed basically our single, after division, the vesicles are single phase. They just contain one of the lipids. So here just the yellow one. And then we feed the green lipids. And we use DNA to mediate the fusion of the green lipids to the yellow lipid. And then we get the phase separated vesicles again. And then the cycle can reproduce basically. So this is now basically, okay, we have a synthetic cell. It can grow, it can divide. Obviously, when are we there? What is still missing? Well, then, of course, you have to ask, okay, what is life, right? What should a synthetic cell be able to do? And actually, there's a lot of debate in the field. You would think it's a simple question, but what is a cell is actually not so easy to answer. So as a working definition, I have to say I go with NASA. NASA says life is self-replication with evolution. So we have self-replication of the compartment. So now we need self-replication of an informational substance, and an informational substance which encodes for the phenotype, for some characteristics of our synthetic cells. So right now we are developing DNA origami, which encodes information and encodes also the shape of our synthetic cells. And I think if we can now combine this with this division mechanism, then I think we'd be pretty close to a system which can really self-replicate and evolve. Of course, one can discuss if this is all that a cell needs. It's a very abstract way of thinking of a cell, of course. But if you think about it, like this is also always something people ask me, like, yeah, isn't this a very abstract and blue skies research that you're doing? And yes, of course it is. Like for me, the inspiration is really the question, is these questions, like, can we build life? What is life? And so on. But for instance, our division mechanism, which is based on the osmolarity change, we found, okay, we can actually also use this as an osmolarity sensor. And if you maybe some of you work with cells or in the fields of biology, then you know that osmolarity sensing in cell culture is actually not possible. You always have to go to an osmometer, freeze the sample, and so on. So basically, we've developed a tool for in situ osmolarity measurements. And this is how I think, without planning for an application of your research and doing really blue sky stuff, quite often useful things come out of it. And of course, these can be patented and so on, and also taken forward in technology. So yeah, this is kind of a pledge for doing this kind of research. Yeah, of course, I want to acknowledge my students. The group has grown a little bit since. They have done all the work. For instance, Yannick, he was doing the division that I showed you of the synthetic cells. Kevin worked a lot on DNA origami structures, DNA nanostructures. And yeah, of course, I thank you for your attention and hope that at some point we'll find the last puzzle piece, the last piece of the puzzle to really build a synthetic cell from scratch. And yeah, with this, I would say my science part is over, and I'd be very happy to discuss with you about other things. First of all, of course, if you have questions to the science, I think we should probably start with that. But then I put down 3 topics. Maybe you have others, and maybe we can choose what we want to talk about, or how do you normally do it? Maybe we start with the scientific questions. Oh yeah, sorry, Ben.
Please, Dr. Göpfrich, show awesome presentation. Very, very, very interesting. I have many questions, but I don't want to hijack this entire meeting. But I'm going to ask you one question that I think people might be interested in. It's sort of a sci-fi question, and I guess in a sense, you're doing some sort of sci-fi research kind of. But the question is, if you can get these cells to divide, you can get these cells to function the way cells do in culture, since they're not derived from any sort of organism. You know, they're not human iPSCs, they're not mouse cells. What do you think they could turn into? Do you think, like, would they form in some sort of organ? Do you have any hypothesis about what they could build?
Yeah, I mean, that's a very interesting question. So far in the field, nobody really talks about evolution, right? But that's actually the most exciting feature, right? I really want to get to a system which can evolve somehow, right? And this, I think, of course, depends on what you put in the end, right? So we have a lot of flexibility and freedom to build features that we like. So of course, first of all, we need to choose according to what we want to select. So I think, for instance, one interesting thing to select for would be swimming speed. So it's quite straightforward to get these compartments to actually swim in solution, to actually move. And if you think about it, if you think about drug delivery applications or so, like swimming micro robots, as you could call them as well, would be quite cool to have. So, okay, we could say, okay, we want something that moves quite fast. So first of all, it should have a shape which allows it to move quite fast. It should have, I don't know, a compartment which is suitable to carry a substance, whatever kind of constraints you want to put. And then, of course, you can select selection criteria to select for these features, right? To really go to, okay, the ones that arrive first will be able to replicate or so, because maybe you provide molecules there for replication or so. That would be how I envision it. I mean,
I was not expecting that answer, but that makes your research even more cool. I think that is really, really interesting.
Thank you.
I have other questions, but let me allow anyone else to ask. You can send them in the chat too if you're feeling shy.
I have a question about the cell mutation thing. So when you're doing research, computational research in biology, like the thing is like biology is not something that's like zeros and ones. It's very like nuanced and like oftentimes inconsistent. So when you're actually doing the research methodology, when you're structuring it, how do you determine confidence intervals, and how do you set like requirements for which experiments are successful and which ones are not?
Yeah, it's an interesting question, because you say biology is not zeros and ones, right? But if you look into the genetic code, it's A, T, C, and G, right? A, T, C, and G you can easily translate into zeros and ones, or zeros and ones you can translate into A, T, Cs, and Gs. It's just that biology is so complex that it's not easy to recognize that it's about zeros and ones, right? Anymore. So I think that's the big advantage that we have. We know all of the ingredients that go in. So actually predicting what a cell does is impossible, right? It's super hard. Like, even though it's the basic component somehow of a living system, right? It's the easiest component that we can study if we don't want to study organisms. Already a single cell is super complex, and all the things that are going on in it. We know exactly, like, we put this many lipids, we put this much DNA, we put this in this sequence of DNA, we put this in this type of lipids. We have a complete control over the composition. So in a way, it's much easier to make predictions, and even our systems are becoming quite complex very quickly. But still, I mean, at least we know everything that's there. So in that sense, that makes it a lot easier. And I think this is also why we as me as someone with a physics background, I like this approach, right? We were trained to build models. And in some way, this is a model for living cell. It's just not as complex. It has some features. It may not replicate all of, or it may not show all of the features, but that's exactly what a model is, right? You focus on the most important ones. So that's how I think about it. Not sure if this answers your question.
It answers it. Thank you.
Thank you. There
is a message or a question in the chat. Were there any ethical complications when you proposed this research idea?
This is a very good question, because no, because if you think about it, like in all these forms that you like when you apply for grants or so, they always ask you, are you going to work with genetically modified organisms? No, I'm not, right? Like all these things, they don't apply to the bottom-up construction of a synthetic cell, because you don't work with anything living, right? And nobody thought about what happens when you make something living, right? Because we're simply not there. So far there's no constraints, and I think that's fair, because any drop of dish soap would destroy anything I make. These are extremely fragile things. They are certainly not well capable of causing any harm. We are simply not there yet. But as soon as you have something which you can call alive, of course you have to think about guidelines, and that's super important. And actually in our research consortia, the bigger ones, there are ethics groups involved, where we discuss exactly these questions, like what happens when we get there. And I think that we should simply set similar guidelines to those we set when we manipulate existing organisms. Regulating it right now, I think, doesn't make much sense, right? It's not very different from, it's non-living matter. It's not very different from working with biomolecules.
So there is another question in the chat. Do you think you would be able to take this research further and try to build a cell that functions as a specific type of cell, for, like, for example, a neuron or a unicellular organism?
Yeah, I think already now we are sometimes putting these synthetic cells into synthetic cell communities, where some of them have a specific functions and others have a different function. So for instance, sender and receiver of a signal or so. So in a very primitive way, you can already differentiate them. This is probably not what you mean, but I'm speaking in abstract terms, right? In abstract terms, you can make two different populations which can somehow interact and communicate with each other. So of course, differentiating an entire organism, I would say we are extremely far away from that, which is also why we're never going to build a I don't know, like a living dog or so from scratch.
I have another question. Two questions actually in one. The first is, so this is a very, as we were talking about, abstract idea. This is an entirely new section of biology and science. So part of my question is, how did you get here? How did you think of this idea? Because it seems so abstract to come to. But the other question is, do you envision this being more so a research tool for modeling cellular processes, or do you think it could also be used as a clinical tool, like the way some like stem cells may be used?
Yeah. So how I got to it, actually, in my PhD, I was building ion channels from DNA. I was making these nanopores from DNA. And I was doing my PhD in a group of Ulrich Kaiser in Cambridge, and he was working on nanopore sensing. So for him, these nanopores were sensors which you could use to detect a current signal and to detect single molecules in solution. For me, ever since I heard the talk in synthetic biology, they were components of synthetic cells, right? And this is somehow where this idea came from, that we can make our own building blocks to make our own cells. This sparked my curiosity, let's say, about this question. Translating it into the clinic, I think it's possible, and it's possible in two steps. So first of all, we are making components for synthetic cells, right? Which directly means we are also making components potentially for living cells, and we are testing them on model systems. But these components could be used for living cells as well. So, for instance, I showed you these plates which can alter the stiffness of the synthetic compartments. Of course, we tested that they can also alter the stiffness of a living cell. So this means now, if you think, for instance, about an immune response, it is known that the ripening of B cells, so the effectiveness of B cells, increases when they are stiffer. So here we can think, okay, can we actually enhance living cells, like use the tools that we make kind of cyborg cells that are enhanced in their functionality with our synthetic components. So I think this will be the first application. The second application in the clinic, and that's far, 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 far away in the future, is okay, once we have the perfect synthetic cell which functions in the environment of a living body, of a living organism, then you could think about it as a micro robot which fulfills a certain function inside that organism. But that's since we are not even there and don't have a synthetic cell, it's super, super difficult to say. But on the way, I think there are things that can be translated into the.
So if anyone has more research questions, feel free to unmute yourself and ask them or put them in the chat. But I think we have about 10 minutes left. Maybe it'd be a good idea to transition into some sort of career-based questions. It's always the best, if you're interested in being a scientist, the best way to learn is to talk to scientists. And Dr. Göpfrich is a fantastic one, as you can tell from her presentation. So does anyone have any questions about her journey or how it may relate to your trajectory, or questions about any of the things she put on the screen? So like choosing a subject, getting a PhD.
A lab, studying in Germany, of course, is always something I can give advice on, but really depends on what interests you.
So if you're going into the research that you're going into, what field would you say is the government giving most funding towards? Which are like the most popular fields?
Okay, I would say never choose your research topic based on that. Maybe I'm giving advice from a German perspective. You have to tell me if this is not applicable to US funding. I think really you have to pick your research topic according to, if you want to stay in science, it's a tricky business anyway. And I think picking a research topic, you should always pick by your own curiosity, and then you can of course phrase it, twist it in different ways, phrase it in different ways. For instance, I do that all the time, right? Then COVID comes up, and then you think, okay, what can synthetic cells do for COVID, right? Then I don't know, the university gets an excellence cluster on synthetic immunology, and you think about, okay, what can I do with knowledge and the expertise that I have for immunology. And then this idea came up with the B cells, like maybe using origami to, but it's always, I think the basis has to be a question that fascinates you. And then of course you can like deviate a little bit from that path, because that's also when collaborations start, right? Like all of the things that I'm doing now, many of them I could not have imagined if I hadn't worked together with other scientists from completely different fields who have the expertise, for instance, in biology that I don't have. So yeah, but first of all, I think what matters is what interests you. And I can't even answer the question. I don't know what German government is putting most money in at the moment, or any other governments. I mean, yeah, of course, there are topics that are now out. Yeah, I would say if you now start to work on combustion engines, if that's your biggest passion, then maybe it will be quite hard. But maybe you're super creative, and I mean, this is what people told Nobel laureate Stefan Hell: don't work on light microscopes, it's over. And then he found a way to make it restart all from yeah. So hard to say if yeah.
Thank you.
There's a question in the chat, which is pretty interesting for your specific path. How difficult was it to choose a certain field to specialize in?
Yeah, for me, it was super difficult after school. Like after school, I was completely confused. I loved it that I was working in that school allowed me to basically look into all the different subjects, sports, so PE, physical, then arts, then literature, then chemistry, physics, biology, all these things. And I really couldn't choose. And I was super unhappy. That's why I chose molecular medicine, because it was at the interface of physics, chemistry, and biology at least. Turned out, no, I went for physics. But then once you specialize in something, then you can go with your interests, right? So right now I'm again in this very, very interdisciplinary field. So once you're an expert in something, it's easy to branch out. It's easy to take your expertise into different directions. I was always really inspired by these big questions, you know, that my parents couldn't answer, the things even as a kid, like when you ask your parents, so Mom, what is life? And then they don't know, then that's exactly the questions that somehow intrigued me or so. And that's where I wanted to get, but it's not obvious how to get there, right? It's not obvious. There's not a subject that's called what is life. So somehow you look for a way to address them in a scientific way.
And how big is your research group currently?
I have 9 students at the moment.
Wow.
So it grew.
Well, that must be really exciting.
It is.
How recently did you start your lab there?
In November a year ago, so a year and a bit now.
Wow, so it is growing pretty quickly. 9 students.
Yeah, it's definitely an exciting transition. I mean, it's crazy.
I had a question. When you were an undergraduate at the University of Nuremberg, were you a physics major?
Yeah.
You were? Oh,
I did physics, yeah. I started studying, maybe we'll talk about it later, I started studying molecular medicine, and I was learning by heart so many things and so many equations. I was like Chinese letters. I was drawing them like I was memorizing everything because I couldn't understand. And then I thought, okay, either I study physics now and then I understand properly what these symbols mean, or, well, I don't know, but I wanted to understand. So then actually I switched from molecular medicine to physics. Then right now I'm kind of in between again. I'm half back.
Your research is very interdisciplinary.
Definitely, yeah. Can be a big frustration over here in Germany. They're very conservative when it comes to that.
Really?
Well, for instance, since my background is in physics, I'm only allowed to be the first PhD advisor of physicists. But of course, my group, I have biotechnologists, biologists, chemists, physicists, everything mixed up. So that means I have to, for every exam, I have to find someone to examinate them because I'm not allowed, unless they are a physicist.
There's a question that just came up. Do you have any advice for undergraduate student that wants to transition from biomedical sciences to neuroscience research. Other than reading research articles, I want to be actively involved in the research side of it and not just learning from reading. And I maybe it'd be best to answer this question in the context of transitioning within any fields.
Yeah. I think reading, contacting the people, just asking for maybe an internship in a lab that's working in the field you're interested in is probably the way to go. And it's really important to say that it's never too late. It's not too late at all after your undergrad. It's probably not even too late after your PhD, because then you're the only person who has this unique skill set of having the skill you learned first and having transition to something completely different. So I would say definitely just look at labs you're interested in and find out if that's really what you want to do. Just give it a try, so to say.
So here's another question. What are some good things to look for in a good PhD program?
Ooh, this is very US specific. We don't have PhD programs. So we do have a few, actually, if you're interested in the topics I spoke about. There's a research school, a PhD school called Max Planck School Matter to Life. So that's really a PhD school focused on these topics. But generally, traditionally, we don't actually have these PhD schools. We just contact supervisors and then we get a salary and then we are not part of them. So I cannot comment. I'm sorry. I have no experience.
That's really interesting. I actually didn't know that. And my next question that I was going to ask is actually, so a question just came in. How was it to study in Cambridge? Was it different from studying in Germany? I was going to ask, what's it like just generally in Europe, being able to sort of bounce around between countries?
Yeah.
In the US, if you leave the US, I would imagine it's much different.
Well, Cambridge is no longer Europe, right? Which is very sad. But no, I mean, so I don't know if you're aware, in Germany, we don't pay any fees. So going to university doesn't cost anything. Doing a PhD, you earn money. And in the UK, it's different. So you have to pay for the university. For PhDs, you get scholarships. Studying in Cambridge is super cool. I'd imagine that the big US schools are a bit similar. It's because people from all over the world come there to do nothing else but their PhDs, and the dedication and the focus and the lack of other, let's say, distractions mean just that your lab mates really become your friends, I would say, because nobody has anything else to do, right? We sat in the lab and built boats from cardboard to row along the cam, and it's just nice because everyone is there together. It's a super international environment. I think it's really amazing. I mean, Europe being able to transition between countries is somehow something that we take for granted, that I grew up with. But on the other hand, if you think about it, the US is just as large, right? So if you now move from California, from New York to California, it's probably a transition that's as big as moving from, I don't know, Germany to Spain or so, I'd imagine, at least. It's difficult to say, but yeah, so study programs in Germany are in English quite often. So this means that you should be able to find something if you're interested in moving abroad. And also there are very nice exchange programs. So if you want to visit the lab just for some time, there's a program called DAAD. It's called the German Academic Exchange Service, where you can very easily get grants on every level as a student, but also as a PhD student, also can get grants to just spend a few months abroad. But also you can at any point email supervisors. Of course, it's probably the same for you guys, right? We don't have bench fees, so we're normally happy to take people for some time if it's not COVID, which makes it harder, of course.
A question about getting like a position at a lab. So what advice would you say if we're reaching out to professors or laboratories that we want to do research in? What advice would you say to optimize our chances of being accepted?
I would say maybe it's obvious for you guys. It's not obvious to German students. Attach your CV. Definitely send a nice email with your CV attached, because then they don't have to ask twice. If you have somebody who is willing to give a reference, especially by phone, you don't attach references necessarily. I'm speaking from, correct me if this is not right for your context, but I would attach like contacts for references, like people that they can call, and yeah, a CV, and that's it, right? That's a nice and friendly email.
Thank you.
All right, well, we're just about out of time. So if anyone has any last minute quick questions, now is your chance. There is a question, there's two questions. One, do international students have to pay tuition when applying to schools in Germany?
No, there's no fees.
Okay, and then there's a very abstract question in here. If the synthetic DNA worked like normal DNA, could you make prehistoric creatures or new animals?
Well, so the DNA we use for DNA origami and so on, it has no genetic meaning. It's too short to have a genetic meaning, actually. So it's just short synthetic pieces of DNA that are used for construction. They have no genetic meaning whatsoever. They are like 20 to 50 bases long. So a gene is like thousands of bases. So that's, yeah, not at all from our side possible, or at least I'm not the person to ask. I mean, yeah, if you could make DNA or sequence these organisms, maybe, I don't know. But yeah, living expenses in Germany are, we have Studiengebühren, they can, yeah, like like 300 euro per semester to cover. At some universities, you can get a free public transport, and then you pay something like, but it's like, it's probably even less than 300 euro per year. But then you get something in return. So it's not so much. Living expenses in Germany, I would say, tend to be cheaper than in the US. So for a flat, you would pay something like a two bedroom flat, you would pay something like, I don't know, maybe $600, $800, $1,000 maybe, depending on where it is. Okay, well, if you do have any questions, feel free to reach out by email also. I have to get home. We have a curfew.
Thank you so much. Your presentation was very, very interesting.
Thank you so much.
Great to learn about. So yeah, thank you for coming.
Cool. Good luck with your experiment over there.
Thank you. Thank you.
Cool.
All right. Thank you all for joining us today. I hope it was informational and educational. I'll see you guys next time.